Okay, يلا. Who wants to read? يلا, who wants to read?
أستاذ أنا ما عندي مشكلة إذا بدك نبلش.
تفضلي يا مريم, يلا.
In the framework of studying immune responses, we do the following experiments. First series of experiments, we perform skin graft between mice A, B, and C of different lines, document 1.
طيب, let's look at the experiments. In experiment 1, skin graft of A from A1 to A1 graft is accepted. يعني لأنه same line. طيب, بعدين let's look at B1. Skin graft of A, mouse B1 of line B after 15 days.
Rejected.
It is rejected. طيب.
Yes.
بعدين second graft of A again, it is rejected after 6 days. First time كانت بعد 15 days. تاني مرة بعد 6 days.
Yes.
Skin graft of line C رجعت rejected by 15 days only. يعني again 15 days.
15 days.
Okay, interpret these experiments. كالعادة, analyze مع significance. We start with the first one.
Yes.
يلا يا مريم, what did you say about the first one?
In document 1, a graft is accepted in mouse A1 when graft of line A was done on mouse A, like mouse of line A.
Okay.
Of line A as well. While graft of B1 was rejected after 15 days when skin graft of line A was done on mouse of line B, mouse B1 of line B.
Okay. فإذا in B1 it is rejected after 15 days.
Yes.
When it is done. While when it is repeated again from the same line A, it is rejected after 6 days only.
Yes.
While skin graft from C is rejected from after 15 days.
Yes.
Okay.
This shows that there should be the same skin graft lines for success.
Okay,
for the
Okay, B1 rejected the same mouse after 15. The graft succeed only between mice of the same line, and the rejection of the graft is faster in the second recognition. يعني second time it will be faster.
Yes.
Okay. طيب. Indicate 2 characteristics of the immune system revealed by these experiments.
The first characteristic of immune system is fighting against foreign bodies وبالتحديد هون في grafting.
Okay. فإذا recognition of the non-self.
Yes. Second is like identifying the type of the graft faster.
فإذا a second response is faster.
Yes.
Okay, I can say كمان here إنه immune system is specific. لأنه in A it did not rejected, in B and from C it is rejected.
Yes.
فإذا عندي هون 3 characteristics. ممكن I choose any 2. Okay, recognition of the non-self, presence of immune memory, and specificity of the immune response. Okay, very good. طيب, clear until now, guys? Any question? Okay, clear. طيب, قاسم وينك؟ محمد, صبحية, please, I need answers, at least in the chat. طيب, Mariam, let's go to the second series of experiments.
Second series of experiments. We graft skin of mouse A into mouse B under different conditions. The experiments done And the obtained results are shown in document 2.
Okay, فإذا now we repeated the grafting from A to B, but now we changed the conditions.
Yes.
Let's see, in experiment 1, what did we do?
In experiment 1 نحن كان عنا بس control mouse B. الgraft is rejected.
طيب why يا مريم we do control experiment?
لأنه نحن بنعرف شو هن الكنترول يعني بنعرف الريزلت تاعهم.
Okay, فإذا الكنترول is to compare with the other experiments.
Yes.
ما هيك لأنه كل ال variables in the control are standard.
Yes.
Okay. طيب.
Second experiment عنا mouse B deprived من thymus.
Okay.
graft is accepted.
تمام.
بالثird experiment عنا irradiated mouse B يعني destruction of bone marrow كمان graft is accepted.
فإذا in the first one graft is rejected بالnormal one. بالsecond وبالthird the graft is accepted. بالsecond ما في thymus. بالthird no bone marrow.
Yes.
Okay, starting from the analysis of document 2, specify the organs involved in a graft rejection. Let's do first analysis.
Yes. In document 2,
the graft is?
First graft is rejected when there is control mouse B.
Okay.
Well, second experiment graft was accepted when there was when the mouse B was deprived of thymus. In experiment 3, graft was also accepted when bone marrow was destructed from the mouse. So the involved organs are the thymus and the bone marrow.
Okay, so to reject the graft, I need the thymus and I need the bone marrow. Okay, skin graft taken from a mouse of line A to mouse B leads to the rejection of the graft. On the other hand, as a control, the graft accepted when it's done on B, deprived of the thymus as in 2, or deprived or irradiated the bone marrow in 3. So these two are the one responsible for the graft rejection. Tamam. Okay, very good. Let's look at the third series of experiment. What we did here.
Yes, we removed cells infected by virus X from a mouse of line A. We incubate the cells with radioactive chromium 51 Cr. This 51 Cr is absorbed and binds to proteins in the cells. After incubation, we wash the cells and culture them with different effector cells obtained from the same mouse A. The
supernatant يعني the remaining liquid are then
collected for measuring the quantity of 51 Cr released by the lyse target cells.
Okay.
Document 3A shows the experiments. With document 3B we'll obtain results.
طيب خليني أعمل explain for this experiment and then we look at the results. Okay, we remove cells infected by virus X from a mouse of line A. يعني now I have cells من line A mouse these cells are infected with virus X. طيب we incubate these cells with radioactive chromium CR51. طيب why radioactive chromium؟ لأنه CR51 is absorbed and binds to the proteins inside the cells. فإذا now the cells are what? Are radiated يعني show radiation لأنه they absorb the CR51 which binds to the proteins inside these cells. Okay. هلا these proteins remain usually inside. They do not get out unless the cell membrane is destroyed. After incubation, we wash these cells. يعني we remove any CR51 still found outside. طيب now المفروض الراديشن يبين بس وين جوات السيلز. Okay. And culture them with different effector cells obtained from the same mouth. يعني we mix these infected cells containing CR51 radiated okay, with different combination of immune cells. تمام. Now, the supernatant are then collected for measuring the quantity of CR51 released by the lyse target cells. Now, if these cells are destroyed, a CR51 will come out in the liquid. فإذا نحن we took the liquid supernatant يعني remaining liquid, we measure the amount of CR51 inside it. لنعرف if these cells are destroyed or not. Document 3A show the experiments that are carried. But 3.1 we did not give them anything. 3.2 macrophage only with these infected cells. 3.3 T4 و T8. 3.4 T4 T8 مع macrophage. Okay, now okay, results. Interpret the obtained results. يلا يا مريم, let us interpret this one. ولا you want me to do it?
لا أستاذ فيني أعمل.
Okay. يلا.
In experiment 3, the quantity of liberated 51CR was 20, which was low, when there was no effector cells.
I see that in 1 and 2 are 20, صح؟
أنا قلتهم كل وحدة لوحدهم.
Okay, أنت فصلتيهم يعني.
Yes.
طيب.
In experiment 3.2, it was also low at 20 AU when the effector cells were only macrophages.
Okay.
Well, in experiment 3.3, the quantity of liberated 51CR was 30 AU medium when the effector cells of the mouse A were lymphocyte T lymphocyte 4 or T8. On the other hand, the quantity of liberated cells, liberated CR, was so high, 250 AU, when cells of the mouse
Mariam, you should
A...
use here the words increase. لأنه it is an analysis.
Yeah, okay. Steve.
Okay, so the amount increased a little in experiment 3.3 to reach 30 AU and increase quickly or sharply to reach 250 AU in experiment 3.4.
Yes.
Okay?
Yes. This indicates that all LT4 and LT8 and macrophages are needed to kill the virus.
Okay, to destroy infected cells.
Yes.
Okay, to destroy the infected cells. فإذا the quantity of CR51 released by the cells lies in a medium deprived of effector cells of mouse of line A and a medium containing macrophage is 20 AU. This quantity increases a little to become 30 AU in a medium containing LT4 and LT8 and reaches to 50 AU in a medium containing LT4 and LT8 and macrophage. So lysis of infected cells require cooperation between LT4, LT8 and macrophages. By referring to the acquired knowledge, يعني something that you memorize, how the effector cells of document 3 intervene in the lysis of infected cells. How effector cells are able to destroy the infected. Okay, مين الeffector cells here that destroy them?
It says T4, T8 or macrophage.
The one that destroy the infected cells, especially.
It says بس نحن مثل ما طلع معنا بال 3.4 إنه بدنا 3
ايه بس I want now to specify the work of each one of them.
استاذ هو أول شيء الماكروفاج هو إنه بيلقطهم بعدين ال T4 إنه activates T8 وال T8 هي destroys them.
اوكي هذا in brief يعني. فإذا read the answer please.
The macrophages digest the free virus recognized as non-self and transform them into peptides and present them on HLA molecules of class 2.
أو MHC2 same.
Yes. These macrophages are thus antigen-presenting cells, APC. These latter migrate towards the lymphatic ganglia where, where they activate the LT4 that secrete IL-2 and activates the LT8 which adheres to the membrane of the target cell and releases preferrin and granzymes that perforate the, the membrane and degrade the DNA of the target cell leading to lysis.
To its lysis. فإذا يا تحسين الماكروفاج cannot destroy the infected cell. Macrophage can destroy the virus, can engulf the virus, and then activating TH or T4, which will activate T8, that will kill infected body cells. Okay, clear?
Yes.
Clear, everyone? Any question? Any question? Okay. طيب, who left? محمد وقاسم left. لا غريب. طيب محمد رجع. اوكي تمام. Okay, very good. طيب, if we don't have any questions, let's move to a new idea in 16. Let's check 16 and see. يلا. طيب, question 16. يلا, who wants to read? محمد أو صبحية؟ يلا محمد أو صبحية، يلا. محمد معنا؟ صبحية معنا؟ طيب, I will start with 16. يلا لحتى محمد أو صبحية يقدروا يشاركوا معنا مشان ما نحطهم absent. Okay, in order to study one of the characteristics of the immune response, experiments shown in document 1 were carried out. Injection of antigen A at time T0 after 7 days, extraction of lymphocyte from lymphatic ganglia. Okay, we culture the cells with or without antigen, 1 without antigen, 2 with antigen A, 3 with antigen B. And we see proliferation of lymphocyte only in culture 2, where we have antigen A, the same one that we injected to the mouse at the beginning. طيب, now write a short text describing the experiment carried on out as well as the results obtained. طيب, ما أنا مشكلة، رجع اطلع منه رجع فوت علي يا محمد. Okay, now I want to describe. In description, I just say what I see without any explanation or analysis. So, this is now question 16. okay, I will say that antigen A is injected into a mouse. 7 days later, cells of the lymphatic ganglia are extracted and put in 3 culture media without antigen in 1, with antigen A in 2, antigen B in 3. We observe the absence of lymphocyte proliferation in 1 and 3, and the proliferation of lymphocyte occurs only in second medium. طب, interpret the obtained result and draw out the characteristic of the studied immune response. If I interpret, I will start from the end. There is a proliferation in medium 2 containing antigen A and lymphocyte taken from lymphatic ganglia of a mouse who was injected with antigen A 7 days ago. Why there is no proliferation in both, okay, culture 1 and 3, which does not contain any antigen as in 1, or antigen B only as in 3. Okay, this shows that what? Proliferation of lymphocytes selected after the first contact with A cannot occur unless the lymphocytes are put again in contact with the same antigen. طب, what I can conclude as a characteristic here of the immune system, I can say that the immune system is what is specific. So, high proliferation of lymphocyte extracted from mouse immunized against antigen A was observed when they are put in culture with this antigen. On the contrary, no proliferation observed when they are alone or contact in antigen B. This implies that proliferation of the lymphocyte selected after the first contact with antigen A cannot occur unless the lymphocytes are put again in contact with the same antigen. Thus, the immune response is specific. Okay, clear this part? Type. In a second experiment, the same steps are repeated without the delay of the 7 days. Yani we did not wait 7 days to take the lymphocyte. We took the lymphocytes directly. The cells of the lymphatic ganglia are directly extracted after the mouse immunization against antigen. The results do not show any proliferation of lymphocytes. فإذا explain the necessity of the 7 days time delay for the lymphocytes proliferation. Okay, so why I need these 7 days? The answer will be that the 7 days is an important time to induce the immune response. I have to wait for the macrophage, okay, necessary to induce the immune response. The macrophage, they need time to phagocyte the antigen and become APC. What we said the meaning of APC? Antigen representing cell that migrate toward the lymphatic ganglia. APC binds to the lymphocyte via their specific receptors and activate them. These, in fact, selected lymphocytes rapidly multiply and proliferate upon a second contact with the same antigen. So that's why we need some time. Okay. In a third experiment, what did we do? We estimate variation in the amount of antigen and the produced antibodies, Ig, during 2 separate injections of the same antigen of an individual. The results are shown in document 2. Type. We have 2 graphs. One for the primary response, one for the secondary response. And the first one, amount of antigen, is the black thick line. Immune complex is 2. Okay, immune complex is 2 with a dotted line. And blue line, which is the free IG, okay, in the blood. الاميون complex اللي هو البايندينج between antigen and antibody. هذا اللي هو الاميون complex. Okay, look guys, if I look to the amount of antigen, what is the difference you see between 1 and 2, between a primary and secondary response? What difference you see? Yalla, tell me, what difference you see between the amount of antigen in a primary and secondary response? What do you notice about this amount?
amount of antigen بأول وحدة ضلت constant، بعدين على اليوم العاشر نزلت، أما secondary response دغري من تاني يوم نزلت لأنه في antibodies.
Okay, فإذا in the first primary response, the amount of antigen remain 1, let's say until day 7. Yes. Then start decreasing to become 0 at day 10. While the amount of antigen in the secondary response decrease directly to 0 after 2 days. Okay. بالنسبة للamount of immune complex يلي هو antibody antigen كمان reach a value, okay, and then decrease to become 0 after how many days? 15 days. بينما بالsecondary response after how many days? Maybe 7 or 8 days. Okay. The amount of antibodies in the blood free in the primary response is a little higher than antigen. In secondary, it goes very high. Okay. Type. Now, if I want to compare and conclude characteristic here.
أستاذ السهم البلو هو لشو؟
سهم البلو is the amount of antibodies in the blood, free antibodies يعني.
Yes.
So now,
okay,
I can say that. No, show poor connection. Please wait. During the first contact with the injected antigen, the antigen amount in the blood decreases starting day 7 to disappear within 10 days. However, during the secondary antigen amount in the blood decreases and disappears after 2 days. More quickly than at the time of the primary response. At the time of the first contact, we inject with the injected antigen, the amount of circulating antibodies in the blood are null and does not appear until the 7th day. They increase to reach a maximum at day 13. On the contrary, during second contact, these antibodies presented as of day 0, they start increase at day 2 and reach maximum at day 7, greater and faster than first contact. Beyond this day, the amount of antibodies in both cases decrease, however, remains high in the second contact. This shows that during the second contact, the antibodies are produced earlier, a greater amount, while elimination of the antigen is faster. Thus, the immune memory favors a faster, stronger, and more lasting response. Okay, فإذا الاميون memory بيعطيني faster, stronger, and more lasting يعني durable response. بيكون أسرع أقوى وبيضل عندي فترة أطول. أوكي بيضل عندي فترة أطول. طيب explain the appearance then disappearance of the immune complex following the antigen injection. أعطيني لشوف appearance disappearance of the immune complex يعني antigen antibody بعد injection of the antigen. Okay, هيدا complex in the primary response it starts appearing from day, let's say 7. Then decrease to 0 after 15 days.
مبين عند day 10.
Sorry?
مبين انه عند day 10 بيبلش.
Day 10 start to decrease again. like كيف عملوا لك اياها dots عند ال 7.
بالprimary response.
بالبرايمري ايه.
اوكي.
It appears من 7 بتوصل ماكسيموم لل 10 وبترجع تنزل للزيرو بال 15.
Yes.
بينما بالسكندري ريسبونس
بين من
they start from day 0 يعني already reach maximum after 2 days وبعدين decrease directly at day 7 or 8. Okay? فإذا appearance is due to what? Neutralization of the antigen by antibodies secreted by plasmocytes. This appearance is due to the opsonization and phagocytosis carried out by macrophages. The macrophage now, okay, what does it do? It will engulf both the antigen antibody after they attach together. They become antigen antibody complex by the process called opsonization. By the process called opsonization. Okay? Clear? Now we are only 2 left. The last one staying will win. Okay, فإذا we see how the amount of antigen is affected, primary secondary response, amount of antigen antibody complex which is بعدين destroyed by macrophage will amount of free antibodies in the blood. How كمان it is affected. Okay, how also it is affected. ممتاز. طيب تمام. تمام كلير ان شاء الله. طيب
استاذ ما بزياده سؤالين؟
هو بزياده صح بس انا بس عم بطلع على 17 انه بناخذه هوم ورك ولا بناخذ شيء with a new idea.
لا استاذ 17 كبير.
اوكي لك عم نشوف ال 18. ال 18 المايس اللي فيه حلوين في اصفر في ازرق وابيض وجراي.
استاذ خلص انا ربحت 3 تحسين.
تحسين طلع؟
ايه.
اوه خلاص لكن يو وين. اوكي طب خلينا لكن ناخذ ال 18 از هوم ورك. رجع تحسين ما طول.
يس.
طيب فاذا وي تيك 18 از هوم ورك. قطع الكهرباء ليش عندك كهرباء يا تحسين نيالك. اوكي اوكي فاذا ليتس رايت داون مشان اللي بده يرجع يفتح الشات بعدين هنقول لهم هوم ورك exercise 18.
استاذ احنا فينا نفتح الشات بعدين خلاص بس نطلع.
ما بيقعد ببين اذا حدا بده يعمل كوبي للانسرز او شيء؟
لا
ما بيختفوا؟
ايه استاذ.
وين بيضلوا لك
سكرنا اللايف
بالجورنال؟
لا اذا عملت ريكورد للسيشن ونزلت على الجورنال بينزلوا كفيديو، بس انه يضلوا الانسرز ما بيضلوا.
ما بيضلهم الا اذا كنا عاملينه كفيديو يعني.
يس.
طيب لكن الافضل يصير بعدين انا الكويستشنز ابعت الانسرز كواتساب.
ايه استاذ هيك بيكون احسن.
هيك بيصيروا بضلهم على طول، يعني امتى ما واحد حب يرجع لهم بلاقيهم، حتى اذا بده يعمل كوبينج يمكن اسهل ما يقعد يحضر الفيديو ويقرب ويرجع فيه.